We put our chart up that represented the animal branch of the tree of life. Last time we had included an outgroup to animals, those choanoflagellates, which are currently not on this picture. They were colonial. They had only one cell type, those collar cells. Sponges we saw last time had multiple cell types. There were no true tissues, but there were multiple cell types, so maybe remember a few of those cell types and what their functions are. In fact, let me get another color just to remind us of what we did last time. There is, and also a reminder, lecture quiz 7 is up on D2L. Post-lab quiz 7 over the stuff that we saw last time, sponges, cnidarians, and platyhelminthes, still on D2L until Thursday. I believe we saw 2 examples of sponges in our lecture last time, the calcium sponges and the glass sponges. In terms of cnidaria, there were 4 classes that we saw. Maybe we could think of a review question of what those 4 classes of cnidarians were. Post-lab quiz has pictures of those classes and examples from them. Platyhelminthes, there were 3 classes of these flatworms that we had to know. So just kind of plugging that post-lab quiz again. I said we would zoom further into the remaining branches of the tree of animals this week. And so today we're going to add 3 more phyla, the annelids, the nematodes, and the arthropods. So maybe before we get into some of the slides, let's add to this diagram the annelids, the nematodes, and the arthropod phyla. And some that we're going to, I guess, talk about on Wednesday. I didn't add anything else to this, but if we look ahead to Wednesday, there's going to be a lab activity over the annelids, the nematodes, and arthropods, the stuff that we introduce in lecture today. So plan on lab activity, another post-lab quiz over annelids, nematodes, and arthropods. And then I think on Wednesday I should have time to finish the other invertebrates so that we can finally have some time to get to the vertebrates. Every time I ask that question about what your 10 favorite animals are, 9 out of those 10 are vertebrates, even though vertebrates, like we said, make up less than 10% of all of the animals. We seem to be focused on those. So let's look at these remaining branches to add a little bit of detail. If we look into this clade of the deuterostomes, we're going to see 2 branches that we're going to investigate further. In fact, one of them we might get into next time, the echinoderms. So echinodermata is going to be a phyla. This term just means spiny skin. So echino is spiny and dermata is skin. These are a phyla that would include things like sea stars or brittle stars. The other branch of the deuterostomes includes the phyla that we belong to, the chordates. So we're going to look at potentially echinoderms next time and save chordates, where we will find all of the vertebrates till I'm sure a week from today. But in addition to the deuterostomes, we saw the other branch that was formerly known as proterostomes. We had broken it down further into this Lophotrochozoan group and ecdysozoans. There were several examples of Lophotrochozoans that we saw from the animal tree in the PowerPoint, but in terms of our examples we'll see in lecture and in lab of the Lophotrochozoans, we can split it into 2 groups. The first is going to be what we talk about today, this phyla of segmented worms called Annelida. So Annelida, we can think of segmented worms, and then the other branch of Lophotrochozoans that we're going to see in lab are the mollusks. So in a smaller letter, I'll just write mollusca. Not too much smaller, but it's I'm trying to show that we're going to focus on the annelids today. We're going to save mollusks and echinoderms for Wednesday. In terms of ecdysozoans, we said this was the clade that had to shed its exoskeleton in order to grow. We'll see 2 branches of this ecdysozoan clade. One of them is going to be our roundworms, the nematodes. So nematoda are going to be the roundworms, and then the last branch is going to be the arthropods, the largest of any of the phyla. And what I wanted to do, kind of like we were doing last time when we identified a particular phyla, like the Cnidarian phyla, we looked at classes within that phyla. We did the same thing with the phyla Platyhelminthes, these flatworms. We looked at 3 classes. Well, we're going to do the same thing for each of these phyla, but I'm about to run out of room. Like I said, we're going to leave off the echinoderms, chordates, and mollusks for a future lecture. And the remaining 3, that gives us the annelids, the nematodes, and the arthropods to discuss today. We doing okay so far? Let's take this, since I just want to be able to write the class examples from each of these phyla on the board. I'm going to erase our to-do list. And let's build our list of classes that we're going to see. And then we can just refer back to this as we click through the slides. So what I really want to do is just continue. Let's take this branch of the proterostomes, because that includes all 3 examples that we're going to see today. So we have Lophotrochozoans, and then Ecdysozoans. That's fine. And each one we're going to further break down into phyla. So each of these are large clade names. We mentioned this Lophotrochozoa is a description of organisms that have this horseshoe-shaped ciliated feeding apparatus at some point in their life cycle. Our examples of Lophotrochozoans are going to be the segmented worms, the annelids, and the mollusks. So more on mollusks next time. Let's take the annelid branch and zoom out a little bit further so that we can see 3, maybe give myself a little more room there. We're going to list 3 classes of annelids. I guess I'll just keep using the same color. The first class of annelids are going to be the oligochaetes. So oligochaetes, polychaetes, that's an A. And the last one is called Hierudinia. Three classes of segmented worms. Maybe let's put those little descriptive things in here. Annelids, these are segmented worms. And of these segmented worms, there are the oligochaetes. Examples are going to include earthworms. Polychaetes, let's just call these marine worms. So we're going to find them in the oceans. And then Hierudinia, this weird sounding group, are going to be the leeches. These are segmented worms that produce a anticoagulant called Hierudin. So the entire class is named after that Hierudin compound that prevents your blood from coagulating and allows these organisms to drink your blood. So three classes of annelids, those segmented worms, but there's one other type of worm that we're going to introduce. And if you're keeping score, we've already seen flatworms. Here are the segmented worms. And now to introduce the final group of worms, that's going to be found in this Ecdysozoan clade. If we look inside the Ecdysozoan clade, there are two phyla that we need to mention. The first one is those roundworms, the nematodes. So nematoda, let's just call these things the roundworms. Most of them are parasitic. There are a few free-living examples. But most of today's PowerPoint is going to be spent talking about the final phyla here, the arthropods. It's the largest phyla, and in fact, it's so large that we need to further divide it into subphyla. So I'm going to say 3 subphyla names. The 3 subphyla to arthropods, and hopefully this screen isn't going to cut us off. The first one is going to be called Cheliceraformes, which is a mouthful. So Cheliceraformes, we're going to see that this means fang-like mouth parts. The second group is a large group that we're going to call Myriapoda. This has been grouped together. That myriapods and insects have recently been, and I know this is grainy, grouped together into one large phyla called Uniramia. Let me write that down. Uniramia, let's just maybe list under here. This is going to be insects and myriapods. I'll explain what myriapods, it's centipedes and millipedes. Cheliceraformes, these are going to be horseshoe crabs and spiders and scorpions. So the horseshoe crab and spiders. Just put a little etc. there because it's a larger group than just the horseshoe crabs or the spiders. Uniramia is the insects and the myriapods, and then the final group, hopefully there's enough room for you to see this, is called the crustaceans. We can describe these crustaceans as marine arthropods. So all of the crabs or the barnacles or the shrimp, anything in the oceans that has an exoskeleton belongs to this crustacean group. So we've got three large subphyla. And that should be good enough for our checkoff list in the slides as we start to click through these. We'll start at the top with the annelids. We'll see examples of these three classes. There aren't individual classes of the nematodes. We'll just recognize them as roundworms. We see a couple of examples. And then we spend some time looking at examples of each of the three subphylas of arthropods. So there's our to-do list. I have erased anything previously, but nobody else has jumped in since then. So we should be ready to attack this. I'm going to kill this light, if that's all right. Is that good for everybody? All right, then let's jump into some of these slides. No lab today, so we should be able to easily get through each of these slides. I'll grab my pointer and we'll march on. So we've got this already drawn on the board in a couple of places. We know that the annelids are part of this Lophotrochozoan group. And if we look inside the annelids, there are going to be three classes that we need to know. And as always, we're going to list some general characteristics of the annelids, and then we're going to look specifically at each of these three different groups. So general characteristics of the annelids, we're going to see, reminding us of where we are in this branch of animals. We're in the branch of bilaterally symmetrical organisms. So we were here in the branch of bilateral symmetry at the end of last lecture. So we're still in the branch of bilateral symmetry. We're looking closely at these Lophotrochozoans. So at some point, these annelids are going to have in their larval stage this trochophore larvae that has that horseshoe-shaped ciliated feeding apparatus. So some of these things are obvious. We can see bilateral symmetry. Maybe it's not obvious, but we can tell from where this thing is located in the tree of animals, it has a lophophore at some point in its life. The other thing that is also obvious from where we are, we mentioned last time the acoelomates that lacked any kind of body cavity. So in the branch of proterostomes, we're going to see some type of coelom, some type of body cavity. The main thing that you're noticing in this picture, well, probably two things. Bilateral symmetry. Synonymous with that is the fact that we've got cephalization. Again, this is all stuff we mentioned last time. Cephalization is the congregation of all those nerve tissues at one end of the organism. So we have the formation of a head and a tail at the other end. Three germ layers, bilateral symmetry, that goes together. The new thing, and maybe the obvious detail that we're seeing from this picture, is segmentation. These annelids are segmented worms, and we can see each one of these segments have these appendages that in this case, really increase the surface area of that organism. This, if I want to give it away, is an example of one of the polychaetes, one of these marine worms. They have a bunch of these little appendages. Something else that's new in terms of bilaterally symmetrical organisms. The only bilaterally symmetrical organism we saw last time, this flatworm, had a gastrovascular cavity. It was one of the things we listed on the lab handout, that this gut sac. Flatworms had it, cnidarians had it. It was a opening that had to act as the mouth and the anus. So nutrients comes in and waste goes out of the same hole. Again, we can see the cnidarians demonstrating this gut sac. But the new advantage that we see with annelids and everything else after this is going to be a complete digestive system where there is a separate entrance for food and a separate exit for waste. So the mouth and the anus are now at separate ends of the organism. Another thing that's unique to annelid, well, the first time we've seen it, it's not completely unique to annelids. We're going to see that annelids, like us, have a closed circulatory system. This just means, if I go to this picture, a closed circulatory system, like we can see on the far side, has all of the blood enclosed in vessels. There's a heart that circulates that fluid, but as long as this fluid is enclosed in vessels, we refer to that fluid as blood. Annelids have this closed circulatory system just like we do. Just as a preview of things that we're going to see when we get to arthropods down here, arthropods, like this grasshopper, are going to have an open circulatory system. This means that the heart circulates fluid that doesn't stay closed in vessels. At some point, that fluid is pumped out into this open chamber and kind of sloshes around in this open chamber. It bathes over the organs. And we're going to see delivers nutrients to the organs and washes away the waste. So the fluid in an open circulatory system is called hemolymph, where the fluid in a closed circulatory system is called blood. So back to this picture, we can see the closed circulatory system. Those are blood vessels with blood running through them. Again, blood is the fluid that is enclosed in these vessels. Just a preview to stuff that we're going to see on Wednesday. There is a lab activity where I'm bringing in some pictures of oligochaete structures, of these earthworm structures. All of these segmented worms, in particular these earthworms, are going to have these, in each segment, these nephron equivalent-like structures. This green tube is basically an individual nephron, and each segment of a worm has its own nephron. If that is a new term, you have nephrons inside each of your kidneys, and it's the job of this nephron. Each nephron is tangled up with a capillary bed, so we can see the red blood vessels, and the nephron is tangled up with that capillary bed, and it's the nephron's job to pull things out of the bloodstream that might be nitrogenous waste. They might be, if the blood gets too acidic, then these nephrons can pull hydrogen ions out of the bloodstream and excrete them in the waste. Each segment has its own little nephron structure and its own little external pore that allows waste to be excreted into the sediment. We're going to see earthworms are really good for gardens because they till up the soil. That allows gas exchange between the roots and the atmosphere. But it also is depositing this nitrogenous waste as it moves through the soil. So they deposit fertilizer as they go. Again, we see segmentation. Each segment has its own specialized structures to cleanse the blood of waste. And there's even a little exit for waste to get out. So those little nephron structures, those mini nephrons, are called metanephridia. Once again, just points out bilateral symmetry. We can see cephalization at one end because that's where all of the nerve tissue is starting to congregate. We refer to this region as the head region, and the other region would be the tail. Anterior versus posterior. Finally, to the individual class examples of the segmented worms. The first class is going to be the oligochaetes. They include the earthworms and the earthworm relatives. The second group are going to be our polychaetes. And the third group we've listed up here is our Hierudinia. The thing that we notice between the first two classes, they have this common ending in keat. What a keat is referring to, if you've ever held one of these earthworms, and I'm going to try and get some that would have ended up as bait, but I might go try and pick some of these things up from a bait shop so that we have them on Wednesday. If you've held one of these earthworms and let it move through your hand, and I know we can't see the details of the picture, but on the bottom side of an earthworm, there are these tiny little protruding structures. They're like nubby fingers, and they use these to move across the surface. They use them to gain traction. Here's a picture of those little nubby-like structures. Those are called, they go by two names. They're called keats, or I grabbed this other picture. It's referring to those keats, those little finger-like structures, as setae. Either term you want to use, describe these little finger-like appendages that give these earthworms traction and allow them to move through the sediment. Their names, oligochaete, just describes that earthworms have relatively few of those keat-like structures, whereas polychaetes, if we can just look at a picture of a polychaete, you can see how many more of those keat-like structures they have sticking off of the sides. In fact, if we could zoom in to one of the segments of a polychaete, you can see each segment have these structures that have a lot of surface area that stick off. So those little bristles that we see, or whisker-looking structures, those are the chaetes. And you can see there are many more of them on a polychaete worm versus an oligochaete. But the other thing to see, in addition to those chaetes that I mentioned, are this elaborate-looking earlobe structure that hangs off of either segment of a polychaete. This structure that I described as an elaborate earlobe is called a parapodia, and it has a lot of surface area, and if we could look more closely, we would see it's filled with a bunch of capillaries. Having a bunch of capillaries and a lot of surface area means that these parapodia can function as a place for gas exchange to occur. So in addition to allowing gas exchange to occur, we're going to see these worms use those parapodia to move around. And that moving water across the surface of the parapodia allows these things to act as gills. It allows gas exchange to take place right across the surface of those parapodia. I'm going to go back just a couple of pictures to this earthworm that we were talking about. Again, oligochaete has a few of those chaetes, so few that you can barely see them here on the picture. Polychaetes, there's plenty of them to see with the naked eye. Just a couple of details about oligochaetes. These earthworms are hermaphroditic. We're going to see structures of them on Wednesday, but these two that are getting together, they have both male and female reproductive parts, so they are capable of making both sperm and egg, but they can't self-fertilize. So the sperm that one makes, it can't fertilize its own eggs, but if it can find a complementary earthworm, then it can share its eggs with the other earthworm and get the sperm from the other earthworm to fertilize its eggs. So we see this reproduction of earthworms occurring here. That structure we're going to call the clitellum. If we just go to this picture here, that clitellum is going to be where some of those reproductive structures are housed. We're going to see external features as well as the internal anatomy. This picture of an earthworm. I even brought out at least that one model so that we can preview it for Wednesday. But I'm going to bring in some actual earthworms so that we can maybe see some of these structures in a little more detail. But what we can see from here, at least, there's a separate mouth, and we didn't make it all the way to the rest of the picture to see the separate anus back there. But I'll bring in on the lab report structures we'll need to see internally. I'll point out at least the closed circulatory system right here. We can see the blood vessels, and there are a few interesting things to point out here in terms of the circulatory system of an earthworm. We can see that unlike us, we have just one heart that pumps and circulates the blood. These earthworms are going to have 5 hearts, and each heart is a ring-like structure that has muscle tissue that just contracts, and it keeps this blood moving through those vessels. So we can count 1, 2, 3, 4, 5 of those heart structures that are ring-like heart structures. Everything else past that, you can see, doesn't have the muscle tissue in the walls of the blood vessels, so it can't pump or circulate blood. But we're going to see closed blood vessels. The other thing to note here that is noteworthy, I'll go back to this picture. Annelids are going to have their nerve cord run down the ventral side of their body. That's weird because we know that our nerve cord runs down the dorsal side of the body. which seems familiar to us, but that's going to be a characteristic unique to chordates, that their nerve cord runs down the dorsal side of the body. Everything else that we're going to see, like these annelids, they have this nerve cord that runs down the ventral side of the body. Okay, so I'll jump over that. We'll leave internal structures for our lab report on Wednesday. I'll go back to this picture of polychaetes. We can, right off the bat, see all of these parapodia, those appendages on each segment that are elaborate, tell us we're looking at a polychaete. And there are all of the chaetes. We can see things like bilateral symmetry, so there's an obvious head region and then a tail region. We can see a bunch of segments. The group gets its name after those chaete-like structures that are abundant on each segment. Those earlobe-like structures that hang off of each segment are called the parapodia. We said those are filled with blood vessels and act like gills on the side of these things. There are the parapodia. And there are those chaetes that I'd mentioned. So those were the marine worms. When we switch gears and look at this final class called Hyrudinea, it describes the leeches. You can still see segmentation, but it has lost all of those chaetes or those setae. It doesn't have any bristle-like appendages that it can use to move through substrate. It doesn't need to, it just needs these attachment points. So when we look at the anatomy of a leech, and I know maybe it's not as fuzzy in the back of the room, but so close to the screen, these smaller words are kind of fuzzy to me, so let me describe these just verbally. We can notice anterior from posterior end on a leech because both ends have these suckers, but the anterior end, where the head is located, it's a smaller sucker, so sometimes called the anterior sucker, versus this posterior sucker, which is more of a base. It's larger than this anterior sucker. So when we compare it over here, this would be the anterior sucker, and then the larger would be the posterior sucker. We can see segments. We don't see any of those little bristle-like appendages. Again, it's named after the substance hyrudin that it produces that prevents the blood from coagulating. Okay, so cross off annelids, those segmented worms. Again, we crossed off flatworms. We've crossed off segmented worms. It leaves only one remaining group of worms, the nematodes, and we recognize those as the roundworms. Most of these roundworms are going to be parasitic, and this comes from the digestive tract of a pig. And you can see it's pretty gross. There's some blood and some fecal matter that's in there, and these roundworms don't care. They explode in such high numbers that it can rupture the digestive tract. In this sketch, you can see those roundworms just full in this intestine. These are those same roundworms that you find in the intestines of mammals. This one again came from a pig, but they could be in humans as well. These roundworms come in male and female forms. You'll notice that the female is larger than the male, and maybe the dead giveaway that we're looking at a male is that the tail end of a male has this little coil to it, a little curve. You can see this little curve. We can see smaller males and the curvy tail. Females are larger and they don't have that characteristic curvy tail. So roundworms, this example that we see here, the genus Ascaris, is one of our examples of a roundworm. And I point that out because there are a couple of these display cases that show roundworms. This one is one that has a male and female Ascaris. You can see the males are smaller and have that little hooked tail. Females are larger and don't have that hooked tail. So we've got one example of a roundworm in lab. The other example is something that we saw in a video when I was talking about those predatory fungus and how they were trapping these nematode worms that were found in the soil. Nematodes, these tiny vinegar eels, are the free-living version of a flatworm. They're not parasitic. You find them everywhere. Here's a little video clip that shows them moving around, and it actually surprisingly points out how numerous these little nematode worms are. I think I have this pulled up, so let me jump over and try and find it. So those are going to be the parasitic roundworms, like the ones we were talking about in the digestive tract of other mammals. This nematode worm is much smaller. It's microscopic. In fact, in the video, the little clip that we saw just before this, it was swimming around in the pond water, and I think, well, I'm just going to jump to this picture right here. You can see it swimming next to one of those water bears. So there's the water bear. There's the nematode, the roundworm. And that's one of those cyanobacteria strips that we can see. That's fine. That's enough of the nematode worm. I just wanted to see that thing moving around. Here's a slide that shows the anatomy of the nematode worm. We don't have to remember all of the structures except for one, and that is this cuticle. That cuticle is what prevents this thing from being able to grow unless it sheds or molts. Molting is another term for shedding this outer cuticle. It is going to act just like an exoskeleton does in this arthropod phyla. Both of them are protective outer structures that have to be shed in order for these organisms to grow. So just pointing out that cuticle, which is why it is part of this ecdysozoan clade, because it sheds or molts in order to grow. Short on the nematode information. So we've crossed off flatworms, we've crossed off segmented worms, and we've crossed off roundworms, and now we are quickly into this arthropod phyla, but we spend some time here because this is the largest of any of the phyla. We said 75% of all animal species come from just this one phyla, so we have to separate it into these three subphyla. And before we tackle the three subphyla, let's look generally at this phyla of arthropods to list some general characteristics they all share. And we know which branch of the animal tree we're still on. We're still on this branch where we're dealing with bilaterally symmetrical organisms that have some type of body cavity. So if they're bilaterally symmetrical, they've got three germ layers. They've got some type of cephalization, so we can see a head and then a tail region. So none of that is new. Again, cephalization, nervous tissue congregated at one end. There is also segmentation that we're going to notice with the arthropods. They're not nearly as segmented as the annelids were, but in each segment of an arthropod, we're going to see that it has specialized for a particular function. We're going to see head, thorax, and abdomen regions of these arthropods that are specialized for different things. Before we get to differences, let's stick on the similarities. The fact that all of these arthropods have an exoskeleton is beneficial because it acts as a coat of armor. This exoskeleton in arthropods is made out of chitin. We said in our discussion of how suits of armor work, they're beneficial because they're protective. But what was the downside? Did we remember what the downside of wearing a coat of armor would be? What was that? Well, well, that is true. If it's an actual, real, living coat of armor, like an exoskeleton, we would need to shed that in order to grow. But if we're like at one of those Renaissance festivals and we just put on a coat of armor, to go into some type of battle, that coat of armor would be helpful because people aren't going to be able to stab us as easily. But if it came to running away from somebody, having this coat of armor, it makes it difficult to move around. So the mobility is the trade-off there. You get increased protection, but your mobility decreases. So again, there's the trade-off with an exoskeleton. The additional trade-off with increasing the mobility of an exoskeleton, what we started to see develop in some of those more advanced suits of armor is those, and not that we have to know advancements in chainmail or any of those things, but have you seen those, I think that's what it's called, the chainmail or whatever it is, where you have some protection, but it allows for the arms to move around a little bit. You have more mobility, but that stuff is not as strong as a steel plate. So we see the same trade-off with these arthropods that have an actual living exoskeleton that they grow. It's rigid, so it makes it difficult to be quick and mobile. And one way to increase your mobility with this exoskeleton is to have a bunch of appendages. And these appendages we can see have joints in them. The The weakest part in this coat of armor are going to be those jointed appendages. Again, the trade-off is that they provide mobility, but they decrease the defense, the protection. In fact, these, let's look at this little time-lapse video real quick. This is one of those cicada bugs that lives underground, and once it comes out of the ground, it does not initially have these wings. It has to crawl up on a tree and shed its exoskeleton and grow its first set of wings. And I say first set of wings, it's the only set of wings. These things are going to live for about 2 weeks. Their job is to find a mate, reproduce, and disperse those offspring. But it can only grow these wings if it can get to a tree stump and crawl to the top of the tree stump. This thing, its original exoskeleton that it had when it came out of the ground was a tough exterior that made it difficult for ants to pierce through this thing. And I don't have a video of this. We're just going to have to use our imagination. But this was several years ago. I wonder if anybody's ever seen the same thing. When these cicada bugs come out during the summertime, it's not uncommon for them to accidentally crawl through like a fire ant bed on their way to the tree. And if this happens to them, the fire ants can swarm these. They're cicada bugs, but remember, initially when they come out of the ground, before they molt like this, that exoskeleton is tough and rigid. So the ants themselves really can't tackle this cicada bug except for in those little joints. Those are the weak spots in the coat of armor. So in this video, and again, it was back when cell phones were terrible, so had I gotten a video, it probably wouldn't have been anything that you could actually see. But I saw a team of fire ants. They were stinging into the little jointed appendages of this cicada bug. And their job, they were trying to paralyze each of those limbs. And when they were able to do that, that cicada bug was stuck and they were able to rip it apart and basically devour it. So exoskeletons are good. The jointed parts are good for mobility, but they're the weak parts, and other insects will take advantage of those and use those to take the organism down. The other thing that's notable about this exoskeleton that we can see from this video is that when these organisms molt, or when they shed, again, molting right there or shedding, that is two terms that describe this ecdysis, this process of shedding their exterior. When arthropods shed this exterior for a few days, well, and let me qualify that. In the case of the cicada bug, it's like a few hours. But in the case of something like this crab that has a tough exoskeleton, when these things molt or shed their exoskeleton, it takes a few days for that new exterior to harden. And that's what makes these things vulnerable to predation. If I go back to this slide, this cicada bug that has just come out of its exoskeleton, the whole thing is soft and is vulnerable to predation. This also reminds me, if you've ever been to one of those seafood restaurants that serves soft shell crabs, has anybody eaten one of those soft shell crab sandwiches? I haven't. It just looks creepy to bite into. But some people love it, and they, at some of these restaurants, will serve a soft-shelled crab. Not that there's a crab that has a soft shell, but what this is a crab that has just molted. It's just shed its exoskeleton. So for a few days, the entire exterior of this crab is kind of soft and pliable. And if you catch a crab during that time and put it in between some pieces of bread with some mayo or whatever they put on there, you can just bite down onto it and make a sandwich out of it. Soft-shelled crabs. Again, they're vulnerable to predation, even by us, right after they molt or shed that exoskeleton. So let's go back to this. I said we'd watch a short little video clip of this thing, and it is extremely short. We can probably see it from there. So there's that cicada bug before it comes out of its exoskeleton. So this hard exterior, really the only soft spots in that were those joints. But now that it has broken from this hard exterior, the entire body is going to be soft and pliable for a few hours. It has to sit out and gulp in some air, and it takes about 30 minutes for these wings to inflate. So now it's ready to go find a mate and disperse those offspring. hold on to that. So we're still on general characteristics of an arthropod. I spent some time talking about that exoskeleton, that coat of armor that has a couple of weak spots like the jointed appendages, and that we have to shed it in order to grow. Arthropods are also going to have a complete digestive system. We saw this last time in annelids, so the mouth and anus are at separate ends of the organism. Something new, even though we saw a picture of it earlier, the open circulatory system. We saw this picture that compared the open circulatory system where fluid is just kind of pumped out into this open chamber and has to slosh around and bathe over those internal organs. Arthropods have this open circulatory system. Here is the complete digestive system that I mentioned, where the mouth is at one end and the anus is at the other. We see open circulatory system. Here is, well, this is a spider, but the structure that I want to point to here are these book lungs. When we look at the book lung, you can see there are a bunch of individual plates. And on each plate there are a bunch of capillaries. This is maybe the example. If you could think of each page in this catalog as being covered in a bunch of capillaries, if I could somehow, I'm going to blow in there to try and separate each of those pages. If we could fan this thing out, that all of that surface area from all of those pages would function as a gill. So book lungs are structures like that, where there's lots of surface area, enough for gas exchange to take place. So going back, gills or book lungs are two modes in which we see gas exchange take place in arthropods. Another way of gas exchange are called these tracheal tubes. Let's look at a picture. Here are these tracheal tubes all along the body of an insect, in this case a grasshopper. There are openings that connect that hollow chamber of the inside of the grasshopper to outside air. These tracheal tubes are connected to these little sacs on the inside. They fill up with air from the outside, and then surrounding these sacs we can see cells of the body. Those cells are producing a bunch of carbon dioxide, and that carbon dioxide has to have a way out. These cells are in need of new oxygen, so they get their gas and get rid of their oxygen through these openings on the side of the organism. These tracheal tubes, they have to be open for this organism to breathe, for these cells to get rid of their waste and to get new oxygen. I point them out just because one of the non-toxic ways to get rid of a wasp nest or anything like that is to get soapy water and to spray these things with soapy water. And what you're doing is you're blocking these tracheal tubes with the soapy film, and that prevents gas exchange from happening between the cells. The cells immediately start to drown in their own waste, and this eventually suffocates these organisms. So if they have to breathe through these little holes, if we plug them, that forces them to drown in their own waste. Maybe kind of a gloomy example, but those are the general characteristics of arthropods. We now zoom in to specific examples of, we're going to visit each of these subphyla to give examples of each one. I'm sure as long as you remember examples of each subphyla, that should help you navigate those quiz questions. We'll start with the first subphyla called Cheliceraformes, which is a weird sounding subphyla, but it describes these fang-like mouth parts. So all horseshoe crabs and all arachnids are going to be spiders, scorpions, and things like ticks and mites. They all have these fang-like mouth parts. Let's look at those real quick. Horseshoe crabs we've seen before. In fact, we've seen some videos of these things moving around on the beach. I think we saw the birds trying to eat their eggs. They moved on to land to lay their eggs at first because there were no hungry mouths, but we see other animals figured that out relatively quickly. When we look at the body of the horseshoe crab, there are two main segments. There is this front segment that we can, obviously there are eyes, so this is what we want to refer to as the head. But we're going to see just two segments. There is this segment that is both the head and the thorax, what we're going to call the cephalothorax. And then the other part back here is the abdomen. There's the abdomen, and then this is our cephalothorax. Then horseshoe crabs are also going to have a tail. This is a relatively small picture, but we can see there's the tail, this what's called a telson. I know that might be kind of pixelated. Telson is the tail. Here is the cephalothorax. and then this region is the abdomen. We're going to see some walking legs underneath there, but I think that I wish this picture was a little bit bigger. We should be able to point out the little fang-like mouth parts that surround the mouth. Those fang-like mouth parts are called the chelicerae. That's where this group gets its name, fang-like mouth parts. Those are the chelicerae. Just remember that these chelicerae are flanked on either side by the first set of appendages called pedipalps. In scorpions and in crabs, these pedipalps have been modified into large pinchers. And then past the pedipalps, everything else is going to be a walking leg. In fact, we see the same makeup in spiders. Spiders, like horseshoe crabs, are cheliceriformes. They have these fang-like mouth parts. So if we look at the sketch of a spider, just a heads up, we're going to do a lab report on Wednesday where we have to know some of the structures of the spider. That's why I have some of these anatomy diagrams. They'll be helpful for Wednesday. What I want to point out now is that when we look at the body of a spider, there is this region that is both the head and the thorax. We call it the cephalothorax. And then the other region is the abdomen. So just like the horseshoe crab, there is a cephalothorax. All of the legs are attached to the thorax region. In this case, it's the cephalothorax. And then there's the abdomen. What we can also see from this picture is that looking from above, we're kind of hovering above the spider, we're looking down at the top of the spider. Those fang-like mouth parts we see are the chelicerae, and then right on either side of the chelicerae, those are the first set of appendages called the pedipalps. Outside of the pedipalps are the walking legs. If we look at the underside of the spider, here we're just flipped the spider so that we're looking at the underside of it. Those are the chelicerae. You can even see the little fangs that are on these mouth parts. We're looking face on at the spider. You can see the chelicerae and then those fangs that are at the bottom side of the chelicerae. There are the fangs. We're looking at the underside of the spider. So chelicerae are these fang-like mouth parts. Surrounding the mouth are these pedipalps. And then outside of the pedipalps, those are the walking legs. Again, we'll see a diagram like this when we get to looking at anatomical structures on spiders on Wednesday. Again, those little small things are the pedipalps, and these much larger structures are the walking legs. We can see 4 pair of walking legs for both the scorpions and the spiders. Here are the pedipalps. Those are not walking legs. Again, in scorpions, these pedipalps have been modified into pinchers. Just like the horseshoe crab, we can see this one body part that is both the head and the thorax, and then here's the abdomen. In scorpions, we can see this is both the head and the thorax region. This is the abdomen, and this is the tail fin with the little stinger. What do I want to... The only reason I kept this, I don't like this picture very well, but it shows that this term cephalothorax is also called a prosoma. Not that we're going to call it a prosoma, we're going to use the term cephalothorax. I just wanted to go back, there was this picture here that used the term prosoma, and it's pointing to this cephalothorax. So going back to this picture, again, prosoma, we will use the term cephalothorax to describe the head and the thorax region. This is the abdomen, and then there's the tail fin with the stinger on it. It's not showing the chelicerae very well, so that's why I added this picture. I thought it was a good picture that shows those fang-like mouth parts. So chelicerae are the fang-like mouth parts. They're flanked on either side by these pedipalps. In this case, we can see the big pinchers. And then in the case of scorpions and spiders, we can see 4 pair of walking legs, the green ones. Those walking legs are attached to the thorax region, so cephalothorax. This is the abdomen, and then here's the tail fin with that stinger. When we move to the next group, this Uniramia, which is our second large subphyla, this is going to include the insects and the myriapods. We're looking right now at the myriapods. Myriapods just mean many segments and many legs. The examples of myriapods are either millipedes or centipedes. Both of them have a bunch of segments and a bunch of walking legs, but here's how we can tell the difference. Notice how the centipedes are they have one pair of legs per segment. 2 legs total per segment. Whereas the millipede here, it's going to have 2 pairs of legs per segment, so 4 legs total per segment. Millipedes are not harmful. They tend to be not harmful. Some of them produce these toxic secretions that in one of these videos we see lemurs getting all messed up on these secretions. It intoxicates them, but it also keeps them free of things like mosquitoes and bugs. That's how it's thought that they eventually got some of these secretions from the millipede. But millipedes are typically herbivores. They're not going to hurt you. Centipedes, on the other hand, these are meat-eating predators where we should see in a video in a second that they actually catch. Some of these things are the size of your forearm, and they can catch bats out of midair. You sent me that video of that Grant, isn't there? The Grant guy that's in Gerald or somewhere north of here. Somewhere north of Georgetown, but he's got a trail cam and there's a cave there, and in that cave there are millipedes that catch bats. There's tons of bats that are coming out of that cave. So these are found here in Texas hunting in caves, and they're found all the way down in South America hunting in the same caves. Here's what I'm talking about. Look at that. Isn't that wild? So you can see how big these things are. They're capable of taking down a bat.
Holding on with its hind legs, it reaches out into their flight path, and almost immediately, it has one.
Crazy. All right, let's get back over to, there was a millipede. Nope, not that one. Here's a millipede. And this is like 13 seconds. I just wanted to show, whoa. We just wanted to show this thing. All the additional legs. We should see 4 legs per segment, 2 pair. There we go. Oh, very short video. All right, back to the slides. So here's where we talked about the myriapods. Those are the millipedes and the centipedes. One is more herbivorous. The other one is a meat eater. The other half of Uniramia are the insects, and insects are sometimes called hexapods because they have 3 pair of walking legs, 6 legs total. So the name hexapods just means 6 legs. And we could count all of these things, whether it's a beetle, whether it's a wasp, whether it's a moth. We can see even a fly, in this case a mosquito. We see 3 pair of walking legs, 6 legs total. Even this Hercules beetle, we can see 3 pairs of walking legs. Something that is noteworthy about these insects, maybe one difference is that everything prior to this, Cheliceraformes and even the crustaceans, we're going to see that they had just 2 body segments. There was the cephalothorax and the abdomen. And now in insects, there's 3 separate segments. There's a head that is separate from the thorax, and then there's the abdomen once again all by itself. So head and thorax have been separated in insects. And on the thorax, that's where we see those 3 pairs of walking legs, and it's also where you might see wings show up. This dragonfly once again shows us 3 pair of walking legs. Those walking legs are attached to the thorax, and we can also see these wings coming off of the same area. Not all insects have wings, but if we're thinking about any of these phyla that have figured out flight, there's only 2. There's the chordates that we belong to. Reptiles, birds, and mammals have even figured out how to fly, but the only other branch to figure out how to take to the skies is one subphyla of the arthropods, the insects. So no crustaceans, no Cheliceraformes, no myriapods have figured out how to fly, but some of the insects have. So we see 3 segments, 3 pairs of walking legs, may or may not be wings. But what we also see in these insects is oftentimes there is a change in the body form from the larval stage to the adult stage. It's what we called complete metamorphosis. You see it in things like a butterfly, where the larval stage is a caterpillar that has a well-developed digestive system. Its job is just to eat and grow. And then it completely changes its body form in the adult stage. It now has well-developed reproductive organs and wings, and its job is to fly around and find a mate and reproduce. There's this big division of labor and a change in body plans in what we call complete metamorphosis. And a lot of insects do this. We saw frogs do this as well. So complete metamorphosis is common in insects, but it's not a rule. There are some insects, like we just saw in the previous slide, that don't go through this drastic change in body plan from their larval stage to their adult stage. In incomplete metamorphosis, we said that the adult stage resembles the larval stage. It's just a little bit larger in its proportions. We have an incomplete metamorphosis. We don't drastically change from our what we would consider larval stage, I guess, to our adult stage. Some insects do. So that's what we described as complete metamorphosis. I will say that that tends to be common in insects. And then we're going to jump to crustaceans, which you also happen to see kind of a complete metamorphosis in some of the crustacean life cycles, which we don't really have to get into a bunch of different crustacean life cycles. But what we need to know about crustaceans is that they're all marine arthropods. So barnacles, this lobster, shrimp could be included in this, hermit crabs, anything that is marine and has this exoskeleton is a crustacean. Here's a short little video of these crabs moving around. I think it's the last one that we're going to see. Let me just make sure. This is interesting, and it's also kind of a review of something that we mentioned last time. We saw those sea anemones last time. This is one of those models of those sea anemones. It was a cnidarian that grew in that polyp form with the mouth and the tentacles facing up. And so that crab scoops up a couple of sea anemones and then just uses these things like Roombas to move around and catch a bunch of things out of the water column and then grab them off the tentacles and eat them. A good example of symbiosis, multiple species working together. In this case, the crab has really kind of taken over the sea anemone. I'm not sure what it gets out of it. That's fine. We don't get to see the rest of that. And the last slide basically is what I was trying to point out as that crab was moving around. We see segmentation in these arthropods. We see bilateral symmetry. You can see cephalization where all of the nerve endings are congregated at one end. We see these antennae, two pair of antennae. We see eyes. We see all of these different appendages that have been manipulated into feeding particles that grab the food and pull it towards the mouth, or these pedipalps that have been modified into large defensive pinchers. We can see, now that we're back into crustaceans, there is a combined head and thorax that we call the cephalothorax, and then there's the abdomen. The walking legs are attached to the cephalothorax, and the abdomen have these tiny little appendages called swimmerets. Again, I didn't bring in our lab activity yet, but on Wednesday we're going to once again quickly look at some sketches of annelids, of nematodes, and of these different types of arthropods so that we're familiar with the body plan. Whether it has two main segments like cephalothorax or abdomen, or in the case of insects we'll see three body segments. There are some other questions in there about the general characteristics of annelids, nematodes, and arthropods that we mentioned. But that takes us all the way through our slides for today.